Hello everyone, welcome to my channel Love of Biology. Today we are going to learn about virus replication. So the replication of the virus. So virus are the obligate parasites. They are very strict parasites. They will depending upon the host organism for their metabolic activities. Intracellular pathogens. They cannot replicate without the mixture and metabolism of the host cell. Viruses are often very specific as to which host and which cells within the host cell they will infect. This feature of a virus makes it specific to one or a few species of the life on earth. So many different types of the virus exist that nearly every living organism has its own set of virus that try to infect its cells. Even the smallest and simplest of the cells, prokaryotes, etc. Bacteriophages are the virus which will infect the bacteria. They have a lytic or the lysogenic cycle, and few viruses are capable of carrying out both. So let us see the lytic cycle. With the lytic phases, bacterial cells are broken open, lysed, that is the lysis, and destroyed after immediate replication of the virion. As soon as the cell is destroyed, the phase progeny can find new host. For example, T4 bacteriophage, which will infect the human intestinal bacteria E. coli. Lytic phases are more suitable for the phase therapy. Some lytic phases undergo a phenomenon known as lysis inhibition, where completed phase progeny will not immediately lyse out of the cell if extracellular phase concentrations are high. In contrast, the lysogenic cycle does not result in the immediate lysing of the host cell. Those phases able to undergo lysogeny, called as temperate phases, their viral genome will integrate into the host DNA and replicate along with it fairly harmlessly, or even become established as a plasmid. The virus remains dormant until host conditions will deteriorate, perhaps due to the depletion of the nutrients. Then the prophases, which will integrate a DNA, become active. At this point, they initiate the reproductive cycle, results in the lysis of the host cell. As the lysogenic cycle allows host cell to continue to survive and reproduce, the virus is reproduced in all of the cell's offsprings. An example for this is the lambda phase of the E. coli. So replication steps for the lytic and the lysogenic cycle are, although the replicative life cycles of the virus differs greatly between the species and categories, there are 6 basic stages. So attachment. Viral proteins on the capsid or phospholipid envelope interact with the specific receptors on the host body surface. This specificity determines the host range. After that, it will penetrate. The process of attachment to the specific receptor can induce conformational change on the envelope that results in the fusion of viral and the cellular membrane. Some DNA virus can also enter the host cell through the receptor mediated endocytosis. So it will penetrate into the host body. Next, uncoating. The viral capsid is removed and degraded by the viral enzymes or host enzymes, releasing that viral genomic nucleic acid into the host cell. Replication. After the viral genome has been uncoated, transcription or translation of the viral genome is initiated. It is the stage of viral replication that differs greatly between the DNA and RNA viruses, which opposite nucleic acid polarity. This process will produce the viral proteins and the genome. Assemblage. After this synthesis of the viral genome, later, after post-transcriptional modifications, viral proteins are packed with newly replicated viral genome into the new virions. They are ready for release from the host cell. This process can also be referred to as maturation. After assemblage, the virus will undergo maturation. So, the new viruses will be formed inside the host cell. When the host cell breaks down, they will be released outside of the host. So, that is the virion release. There are 2 methods of virion release, lysis or budding. Lysis results in the death of the infected host cell. So, these type of viruses are referred to as cytolytic ones, which is the example is variola. That is a smallpox virus. Enveloped viruses such as influenza virus are typically released from the host cell by budding. In this cell process, the results in the viral phospholipid envelope. This type of virus do not usually kill the infected cell and are termed cytopathic viruses. After release, some virus proteins remain within the host cell membrane, which acts as a potential targets for circulating antibodies. Residual viral proteins that remain within the cytoplasm of the host cell can be processed and presented at the cell surface on the MHC class 1 molecules, which are the immune cells, where they are recognized by the T cells. So T cells will recognize these things and they will be released. So these are the replication steps. First, it will infect the virus particle in both lytic and the lysogenic cycle. After infection, so they will penetrate into and this will continues. Thank you for watching. Please subscribe my